This is an introduction to pharmacogenetics, its utility, and implementation. It is well recognized that many medications exhibit a wide inter-individual variability in their efficacy. Pharmacogenomics is the study of these inherited genetic differences in drugs' conversion pathways and investigates how genetic variations affect an individual's response to a medication. The goal of pharmacogenetic analysis is to adjust a medication regimen that maximizes benefits and minimizes risks of side effects. Pharmacogenomics enables predicting drug efficacy, identifying optimal treatment, preventing adverse effects, and reducing trial and error prescribing. Pharmacogenomics was originally pioneered for personalized prescribing in cancer treatment or oncology. Soon it was being utilized to identify patients who were more likely to benefit from specific common medications or less applied for specific common medications. Pharmacogenomics is currently being implemented clinically for chronic diseases. Here is a brief historical overview of pharmacogenomics. In the 1970s, even though much progress had been made in the treatment of breast cancer, physicians didn't understand why some patients responded better to the treatment than others. Then in the 1980s, there was a breakthrough discovery. Scientists discovered the HER2 receptor, a cell growth regulator gene that's overexpressed in some breast cancer patients. These patients had a normal expression of the HER2 receptors. The HER2 receptors drives cancer cells to divide more rapidly. Patients with the HER2 overexpression faced a grim reality that their cancer would not respond well to conventional chemotherapy and reoccur earlier, with survival rates were much lower. This type of breast cancer is now known as HER2 positive. A monoclonal antibody called Herceptin developed to block the HER2 receptor's ability to drive cancer cells to divide rapidly, showing the progression of HER2 positive breast cancers. The FDA approved Herceptin in 1998, making Herceptin the first ever personalized treatment for breast cancer. Today, patients diagnosed with breast cancer undergo genetic testing for HER2 overexpression. We now know that around 20 to 25% of all breast cancer patients are HER2 positive. Treating these patients with Herceptin means that a HER2 positive diagnosis no longer means a lower survival rate. The success of Herceptin has accelerated the introduction of personalized medicine in the pharmacogenomics in other diseases. Today, up to 50% of all new medications have a companion diagnosis to improve accuracy and safety of the treatment. As you can see, the history of pharmacogenomics is quite extensive, and whether you're practicing in oncology or other specialties throughout the nation, pharmacogenomics should always be a major part of what you do for you and your patients. In summary, drug response varies from person to person. Pharmacogenomics can predict an individual's response to medications and help physicians and pharmacists personalize medication regimens. The benefits of pharmacogenomics are recognized worldwide, and many leading healthcare systems have incorporated pharmacogenomics in their testing in clinical practice. Leading healthcare institutions are implementing pharmacogenomics testing. The list includes the Pharmacy Association of British Columbia, the Montreal Heart Association, the Mayo Clinic, CAMH in Toronto, St. Jude's Hospital, the Vanderbilt Medical Center, and the University of Florida Health System. These institutions have implemented various pharmacogenomics programs covering a wide range of therapeutic areas, including pain, cardiovascular, mental health, oncology, and immunology, just to name a few. Here are some exciting results. Mayo Clinic successfully incorporated pharmacogenomics prescribing and was able to utilize their EMR. St. Jude's found out that more than 1/5 of their patients tested had one or more high-risk variations in enzymes which metabolize medications, leading to dose changes. Vanderbilt predicted that their program helped avoid close to 400 significant adverse drug events in 5 years. Overall implementation of pharmacogenomics helped improve patient safety and reduced overall healthcare costs. Thank you for sharing your valuable time with me today. Learn more how you too can embrace this revolutionary testing in your practice. Join us saving lives one test at a time.